Our next speaker is Dr. Kiichiro Tomoda, research investigator, Gladstone Institute, and Program-Specific Research Center Associate Professor at Sailer, Kyoto University. Dr. Tomoda was the first postdoc at Yamanaka Lab at Gladstone Institute, and he recently joined back to lead the on-site laboratory. Today, he will be introducing their latest research with the title NAT1-Mediated Protein Translation in Pluripotent Stem Cells. Dr. Tomoda, please start.
Thank you, Dr. Saito, for kind introduction. Today, I'm going to talk about our favorite gene, NAT1, or EIF4G2. I will use the word NAT1 instead of EIF4G2 in my talk today. So I will introduce protein translation initiation, briefly introduce protein translation initiation to you because NAT1 may be involved in this process. Then I will share results of our progress led by Dr. Kazu Takahashi and reported last year. This is a snapshot of protein translation initiation. This process is very complex and many proteins and RNAs are involved in this process. I will not explain the entire process today because of time limitations. However, one of the important take-home message is here. It is very crucial for the target messenger RNA to form this circle structure for its efficient translation. And the protein called 4G1, EIF4G1, play an important role in making this structure by scaffolding EIF4E, which bind to the 5' cap of messenger RNA, and PABP, or poly-A binding protein, which bind to poly-A tail. The other take-home message is, although this picture is a general view of protein translation initiation, only roughly 65% of translation in a cell depends on this mechanism. In other words, roughly 35% depends on the other mechanisms, and this dependency may change depending on a cell type and the situation in a cell. Here is our favorite gene, NAT1. As you heard from Dr. Yamanaka, he discovered NAT1 in the late 90s and reported NAT1 is essential in mouse early development and in mouse embryonic stem cells. What has intrigued us is NAT1 is very similar to EIF4G1, but lacks the very important functional domains, two domains, through which 4G1 can bind to EI4E and PABP. So NAT1 cannot bind to EI4E and PABP. What this means is NAT1 cannot form this important structure to enhance translation. However, NAT1 seems to play an important role in protein translation. We have been exploring NAT1 functions in pluripotent stem cells, particularly in human iPS cells. To do this, we made a NAT1 conditional knockout human iPSC line. As you can see here, in this line, Adding dogs in a culture medium sustained NAT1 protein expression, and removing dogs from the culture medium suppress the expression. When we knocked out NAT1 in the iPS cells, we immediately observed iPS cells just die or differentiated. This observation was consistent with the gene expression. As you can see here, the pluripotent marker genes are downregulated, but the differentiated marker genes are significantly induced in NAT1 knockout cells. However, we noticed that even in the differentiated cells, we didn't observe the neuroectoderm marker gene induction in the NAT1 knockout cells. To dig this phenotype, we spontaneously differentiated iPS cells into 3 germ layers and found NAT1 knockout cells could differentiate to endoderm and mesoderm lineages, but could not differentiate into neuroectoderm lineage. So NAT1 is required for the differentiation into neuroectoderm lineage. This observation became more obvious when we forced differentiated iPS cells to neuroprogenitor cells. So we couldn't get any neuroprogenitor cells without NAT1. In summary, NAT1 knockout induced the differentiation of human iPS cells. So NAT1 is essential to maintain human iPS cells. Interestingly, however, NAT1 seemed to prevent human iPS cells from neuroectoderm differentiation. What's the mechanism behind this? Previously, we proposed NAT1 forms this kind of protein complex and actively participate in protein translation initiation. So we hypothesized NAT1 regulates translation for specific proteins to maintain human iPS cells. To test this hypothesis, we looked for target messenger RNA which bind to NAT1 or NAT1 containing complex. Actually, we found several hundreds of messenger RNA that bind to NAT1 in human iPS cells or in mouse epistem cells. Mouse epistem cells are one of mouse pluripotent stem cell types and very similar to human iPS cells. Because the same differentiation defect phenotype was observed in mouse epistem cells, similar to human iPS cells, we focused on these 50 genes to analyze further. I will focus on one of them, one of the 50 genes called TUT7 in my talk today. TUT7 is an enzyme which adds origins to messenger RNA. I will explain a little bit more later. First, we checked NAT1 knockout affects TUT7 expression. As you can see here, TUT7 messenger RNA did not change without NAT1. However, TUT7 protein expression was dramatically reduced when we knocked out NAT1. As a control, we checked TUT4 expression. TUT4 is a TUT7 family member and its expression did not change without NAT1. Thus, NAT1 specifically affects TUT7 protein expression. Next, we introduce TUT7 genes into NAT1 knockout cells. When we introduced TUT7 ORF only or ORF plus 3' UTR genes into the knockout cells, TUT7 proteins are efficiently expressed even without NAT1. However, when we transduced full-length or 5' UTR plus ORF gene into the cells, TUT7 proteins was not expressed well in the absence of NAT1. Conversely, all genes transduced are well transcribed. These results suggest NAT1 affects TUT7 protein expression at a post-transcription level, and the 5' UTR of TUT7 messenger RNA seem to have a region which suppresses TUT7 protein translation, which NAT1 released. Finally, we fractionated iPS cell lysates, got ribosomal fractions, and asked which fractions have TUT7 messenger RNA. As you can see here, when NAT1 was expressed in the cell, TUT7 messenger RNA is in heavy polysome fractions, suggesting TUT7 messenger RNA is well translated. However, when we knocked out NAT1 in the cell, TUT7 messenger RNA is mainly in light polysome fractions or monosome fractions, suggesting TUT7 messenger RNA was not well translated. In summary, NAT1 affects TUT7 protein expression by regulating its translation. So TUT7 is a target of NAT1. Then the question is whether TUT7 is responsible for the NAT1 knockout phenotypes. As I explained a little bit, TUT7 is an enzyme which adds to a short poly-A tail to induce degradation of this messenger RNA. So TUT7 is involved in messenger RNA degradation. However, TUT7 functions in pluripotent stem cells are largely unknown. So we knocked down TUT7 shown at bottom row and found knockdown of TUT7 cause same phenotype as NAT1 knockout cells. So we couldn't get neural ectoderm cells from TUT7 knockdown iPS cells. Then next question is how NAT1 and TUT7 maintain the iPS cell differentiation ability. So since TUT7 involves in messenger RNA degradation, we hypothesize upregulated gene of TUT7 target messenger RNA cause neural differentiation defect. To test this hypothesis, we checked the global gene expression and found human endogenous retrovirus or HERV-H elements are significantly induced in NAT1 KO cells. This result is quite striking to us because we previously reported iPS cell lines that have high HERB-H expression shows neural differentiation defect. So to directly test HERB-H involvement in the NAT1 knockout phenotype, we suppress HERB-H expression in NAT1 KO cell, knockout cells. When we knocked down HERB-H, shown in the bottom, NAT1 knockout cells could differentiate into neural lineages. We also check if TUT7 involved in HERB-H expression and found not only NAT1 knockdown, but also TUT7 knockdown induced HERB-H expressions. Furthermore, we checked the uridylation of HERB-H elements and found NAT1 knockdown and TUT7 knockdown reduce the level. Finally, we checked the HERB-H transcription level, and we found the nascent transcript of HERB-H, shown in red, did not change without NAT1, although the total expression of HERB-H dramatically induced, suggesting this induction is at a post-transcription level and maybe inhibition of the degradation dependent. In conclusion, we found NAT1 is required for the self-renewal and neural differentiation of human iPS cells. Loss of NAT1 increases HERB-H expression, which inhibits the neural differentiation. Also, we identified TUT7 is an important target of NAT1. NAT1 promotes TUT7 protein translation, and TUT7 knockdown phenocopies some of the NAT1 knockout phenotypes. I will conclude my talks with future plans and our history. The study I showed you today has given us some of many important questions shown here, some of them shown here. We will address these questions, and we envision figuring out or addressing these questions make it possible to control iPS cells by reprogramming translation, protein translation. That is our goal. This is our history. So actually, our laboratory here at Gladstone started in 2007. Since then, we have explored human iPS generation, iPS characterization, and reprogramming mechanisms with close and great collaborations with many researchers, not only at Gladstone, but also at UCSF. As you can see here, our research purposes are evolved. Although our research purposes are evolved, we will continue the great collaborations and further stimulate collaborations between CYRA, Kyoto University, Gladstone, and UCSF, or even many researchers in the Bay Area. With this, I would like to thank many people, many mentors and collaborators listed here. Particularly, I would like to thank administrations at CYRA, Gladstone, and Kyoto University for making this exciting program happen. Thank you for your attention, and I'm happy to take your questions if you may have.
Okay, thank you. Thank you, Dr. Tomoda. So now we are taking some questions. So please post your questions in the Q&A box if you have. So the first question I will read. So does DOX treatment for the inducible NAT1 knockout lead to any toxicity in iPSCs?
Yeah, so DOX may have some effects, unwanted effects. So we have to optimize concentration of DOX added. So, and also we need appropriate control to check the phenotype of NAT1 knockout by DOX withdrawal or addition.
Okay, thank you. So I'd like to ask your future plans. So you mentioned about how NAT1 can control protein translations. And also I'm wondering how NAT1 can control the protein translation level of this TUT7 also. Do you further explain about your future plan about this direction?
So we may need in vitro translation system to reconstitute protein translation initiation by NAT1. Or alternatively, we have to purify NAT1 containing complexes or knock down its components to see if we saw the same phenotype as NAT1 knockout. Those research would narrow down the mechanisms.
I see. Thank you. So the next question is, was there any cell degradation when removing the DOX treatment?
If I understand correctly, cell death, you maybe yes, we saw many cell deaths when we knocked out NAT1, and the remaining cells are differentiated into mesoderm or endoderm cells.
I see. Okay. So the question is how self-renewal ability and differentiation ability could coexist? And also, have you done in vivo study for it?
Yeah, so we are planning to do an in vivo study, making a mouse model to knock out or something in the mouse to see what happened. Also, so Dr. Yamanaga already observed early embryonic lethality in NAT1 knockout mouse. Also, sorry, okay. Oh, so the question is, what's the relationship between self-renewal and pluripotency? I think that's a really good question. I don't know the good answer yet. So, but hopefully our research will shed light on this question.
Okay. And the next question is, do you have an idea what percentage of differentiation is pre versus post-translational regulations? Or what do you expect is likely breakdown?
That's a very interesting question. So we don't know, but genes should be translated. So post-transcription mechanism may be very important, but we still do not know what the importance of this regulation is.
Okay, thank you. And next question is, could you please explain why NAT1 fails to induce the iPS differentiation into neuroectoderm?
Yeah, so so in my talk, I explained NAT1 knockout induce HERB-H expression. So HERB, and if we knock down HERB-H expression in NAT1 knockout cells, iPS cells, those iPS cells could differentiate into neuroectodermal lineage. So it seems like HERB-H inhibits neuroectodermal differentiation. So we don't, but we don't know how HERB-H inhibits neural differentiation. There is some literature to explain this. One of them, HERB-H maybe affect chromatin structure, and that may inhibit neural differentiation, but we don't know the actual answer yet.
Okay, thank you. Okay, so next question is, as Dr. Yamanaka talked, NAT1 is required for mouse ESC differentiation, not cell survival. So in human, NAT1 knockout affected both cell survival and differentiation into meso-endoderm. So did you test NAT1 knockout in human naive PhD?
Sorry.
Yeah, please first.
Yeah, I think that's a good question. So because of time limitations, I didn't explain. Human iPS cells are at a little bit different pluripotent state, different from what in the mouse pluripotent ES cells. So then when we knock out iPS cells in human naive cells, naive pluripotent stem cells, we observed the same phenotype as mouse ES cells. So I think this answer your question.
Mm-hmm. And yeah, following that, and do you imagine NAT1 have some functions in peri-implantation development where pluripotency change from naive to prime?
Yeah, I think so. We don't know when NAT1 knockout mouse embryo die, but I expect maybe around the pre or post implantation, NAT1 knockout mice die. So where maybe NAT1 play an important role.